All right. So as we continue our sort of review of what you've covered in your undergraduate careers, we're going to talk about membrane, proteins, DNA, RNA, and receptors, right? So this is sort of like cell bio in a very rapid amount of time. So those are our objectives, the things I want you to take out of this. Cellular anatomy. This should be familiar, right? You've had a number of courses in your life where these things were talked about. All I've done here really is make a kind of anatomy cheat sheet for the cell, the anatomy of the cell. So we have our membrane. Don't ever underestimate the importance of the cell membrane. It may sound a little philosophical, but without the cell membrane, life as we know it would not exist. Believe it or not, it's just that simple. In order for life to happen, there has to be two environments. There has to be an internal environment, and that environment has to be different than the external environment. Well, how do you maintain difference between one environment and another? Well, you need a boundary of some kind. You need a container. That's the cell membrane. So without a cell membrane, we don't have any way to separate internal from external. So even though it's a simple structure, we're going to spend a fair bit of time over the course of this semester talking about things that happen at the membrane. Because ultimately, it's the membrane that's going to determine how that cell, how that tissue affects the whole body, right? So cell membrane, big deal. Centriole, important to division, important to mitosis and meiosis. Those are all cell biology topics. We ain't going to talk about that stuff, okay? So just know they exist. Golgi apparatus, protein packaging and transport. This is like what I want you to remember about these organelles, okay, from your past. So the Golgi apparatus is all about turning long chains of amino acids into functioning proteins, right? So we have that. Endoplasmic reticulum plays a role in that as well, in getting proteins into their final active form. We have a rough ER and a smooth ER. Rough ER is where proteins are made, right? That's where the Lego blocks are put together in their long chains. It's in the Golgi apparatus and the smooth endoplasmic reticulum that those chains are put into their final functional form, right? The ribosome is what makes the rough ER rough, okay? The ribosome, protein construction factory. It turns a DNA template through a messenger RNA into a protein that has the right amino acid sequence. That's all a ribosome does. A lysosome allows for the cell to break down materials that it has brought in. We'll visit some examples of that in the gastrointestinal tract. Peroxisomes are kind of a specialized lysosome. In a peroxisome, we have peroxides like hydrogen peroxide and other peroxides that provide for oxidative degradation of substances. So for example, the heme molecule is difficult to break down. We need peroxisomes in order to do that. The mitochondria, we do talk a fair bit about in our metabolism and energetics chapter. This is the site of oxidative phosphorylation. The current theory is mitochondria used to be independently living bacteria that took up residence inside of eukaryotic cells. And, wow, what a match made in heaven that was, right? Because we get the benefit of a very efficient and effective ATP producer in the mitochondria. Cells do have a skeleton, even though I think we don't appreciate that as much as it probably really exists, because when we look at the microscope at cells, we don't see the skeleton, but cells are not in the shape they're in by happenstance, right? They put themselves in the shape that they want to be in, and that's the cytoskeleton at work. The nucleus is the site where DNA is stored. For our purposes in this class, it's also the site where DNA is turned on and turned off. Okay, so one of the themes of this class is that cellular function has to change as the needs of the body change, right? And one of the ways that that occurs is by turning genes on and off. We'll talk a lot about that in the hormonal system. And then cilia. Not all cells have cilia, but some do. What cilia does is they create movement external to the cell. So, for example, in the respiratory tract, in the cells that line your trachea and your respiratory passages, those cells have little hairs that beat in a uniform fashion. And by moving all the time, they're moving mucus up and out of the pulmonary system. Works like a cleaning system, right? You know, even though we don't like to have to blow stuff out of our nose or cough stuff out, it is in fact the way the body keeps the respiratory tract nice and clean, right? So we'll see a couple of examples of cilia as we go. Cells do stuff. Some of the things they do. Pinocytosis, right? That's cell drinking, as you probably learned about it. And this is where we get a little bit of a pinch of membrane. That's this top one, is pinocytosis. We get a little bit of membrane that pinches in, and then whatever was outside the cell is brought in through pinocytosis. Phagocytosis is a similar process, but it involves larger bits, right? Pinocytosis is more bringing in fluid. Phagocytosis is more bringing in solids. And then remember that no part of a cell is as old as that cell is. Just like in you as an organism, there's no part of you that's as old as you are, right? Because things are being turned over all the time. They're being replaced all the time. So in a given cell, you know, an organelle has a lifetime. When that lifetime is over, it's digested and a new one is made, right? You know, I guess you could argue that the nucleus, that doesn't, the nucleus isn't remade, but most of the cellular organelles are. So that's what we call autophagy, eating oneself. It's a lovely word, isn't it? Okay, so one of the cellular activities we talk most about is protein synthesis, or at least synthesis in general. Many cells of the body produce something that is released into their external environment. You know, so sweat on the skin, saliva, the GI tract is full of secretion. The pancreas, that's pretty much its only job, is to secrete. The stomach has a number of secretions, the liver, and so on. So how does this production of molecules occur? Well, for a protein secretion, we're in this plan right here. So we have rough endoplasmic reticulum here. This is where the proteins are made, right? Those proteins are formed, moved to the smooth endoplasmic reticulum where they start to take on their final shape, through the Golgi apparatus where they end up in their final shape, and then they're shuttled to the membrane using cytoskeleton bits. And then what was produced way back here is finally released into the bloodstream or into the GI tract or wherever we are. This same process can occur for lipids. We skip the rough endoplasmic reticulum part, but smooth ER produces lipids. A similar process occurs for carbohydrates as well. So cellular synthesis, creation of stuff, usually involves multiple organelles working together. And let's see, what was I trying to get across of here? ATP synthesis is ultimately oxidative phosphorylation, so we'll come back to that one. All right. So if a cell has to change its behavior according to what's going on in the body, well, we need a system for that. And genes, we continue to learn more about genes. The landscape of understanding about genetics and genomics continues to change, every year. What we do know is there are a lot more genes than any given cell ever uses, right? So if you think of DNA as a library, most of the books in the library are never checked out, right? They just sit there. Well, gene activation is getting one of those library books, one of those protein templates, and getting it activated so that the cell actually produces that. We have a number of ways of turning genes on and off. Probably the simplest to understand is the steroid hormones. The steroid hormones are produced in different glands of the body, released into the blood. They then cross through the cell membrane and through the nuclear membrane and affect nuclear transcription. So, for example, estrogen, when it enters certain cells, will activate whole new metabolic pathways, right? So we turn a gene on, that gene is then transcribed into RNA, that RNA leaves the nucleus, goes out into the cytoplasm, finds a ribosome, and that RNA template is then produced into a protein. And then eventually that protein has to go through its structural changes until it actually functions like it's supposed to. Take my last slide and then we'll do another Kahoot here. Okay, so, what do cells do? Cell division, i.e. mitosis, after the fetal stage, all right, when we go from fertilized egg to embryo to fetus, we have differentiation that occurs. In other words, the mother cell, when it divides, the daughters are not exactly the same as the mother, right? It has to be like that because you got to get heart cells and lung cells and skin cells and bone cells. So you started with one cell. They have to differentiate. After that initial development and initial construction of tissues, most cell division is cloning. In other words, the daughter cells are the same as the mother cell. Now, we'll see some examples where that isn't the case, but for the most part, that is true. In the embryo, we get that differentiation so that we can get the different tissues started. We get some differentiation as adults too. So, for example, in the blood cell making system, right? You have a stem cell that produces lots of different kinds of blood cells. Okay, so more on that when we get there. And then last bit I want to talk about cell lifestyle is apoptosis or apoptosis, depending on who you ask again. Lots of different pronunciations for things in science. And I think in part it comes from the fact that people read more science than they talk, right? So pronunciations can get a little weird. Who learned it as apoptosis? Who learned it as apoptosis? Nobody. Okay. Well, I learned it as apoptosis too. Okay, so apoptosis, this is where healthy cells die on purpose. Cell suicide is what this is. Now, it's the best way for a cell to die. And remember I said earlier, none of your cells are as old as you are, right? With the exception of maybe a few nervous system cells. So if that's the case, we have cells that are dying and cells that are being made every day. Well, the most efficient way for the body to deal with this is to have a cell that's dying to pretty much clean itself up and destroy all of its bits before it goes away. Because if a normal healthy cell were just to give up the ghost and die, well, it's going to leave junk, right? It's going to leave little bits of organelle, bits of endoplasmic reticulum, proteins everywhere. We don't want that. Then we get a bunch of inflammation. Apoptosis, this is like a switch where the cell starts to destroy itself. It stops making new stuff. And then by the time it's done, it just kind of involutes and there's very little debris left, right? So apoptosis is the way we want cells to die. Now, in pathology, trauma, infection, oxygen, nutrient starvation, problems with homeostasis, apoptosis is not what we get, right? We instead get necrosis or other inflammatory or inflammation-causing forms of death, and then we get more trouble. You're going to learn much more about that in MedPath, right? If you haven't started talking about it already. Okay, let's do another Kahoot. Animal quiz, where are wallabies found in the world? Australia, very good. How about puffins? Yep. New Zealand, Antarctica. I think there are some there. And Yetis? Washington, right? That's a Bigfoot, in case you didn't know that. Okay. Got 4 questions. A stack of membrane pancakes. This organelle helps proteins achieve their final form. Very good. See, now you made a cell biology teacher very proud. Yeah, that's the Golgi apparatus. Oh, high scores. You guys are quick. All right, this set of membrane-associated proteins plays a role in pinocytosis. Oh, something I didn't talk about in class. Ho-ho! See, and that's what happens when I don't point it out in class. That's clathrin. If you look back at that slide that has pinocytosis on it, you'll see that clathrin just sits right underneath the membrane, and it plays a role in getting the membrane to divot down and make a not a centriole, kind of like a lysosome to pull in. At least you guessed very randomly or very evenly. The polite dragon came up on that one. Okay, 2 more. Which of these plays a major role, that should be, in the determination of cellular function? Read all the answers. Good, all of these. Okay, another tip from me. What I try to do in this first, before our first test, is give you hints about test taking in the PA program. All of these is not always correct. All right, so there's point number 1. Be sure that you can convince yourself that they're all true. We don't have a lot of those on our tests, but they are not always true, and we see students make that mistake. Oh, polite dragon holding the lead. Mitosis requires which of the following? DNA replication, best answer there. I suppose if you really wanted to get into the nuts and bolts of it, you could make the argument that you got to have RNA to make it happen in the first place to produce the proteins that lead to cell mitosis. But we have to replicate the DNA because we're about to get 2 cells, right? We get a lot of protein creation, not destruction. And we don't break down the chromosomes, we organize them, right? So that they can be split apart into those 2 sections. All right, and that is it for me today. I will get the movies working, and we will do the movies next time. And we'll start in on, we'll finish that chapter and probably get in a little bit into the next one too. Our winner was the melodic newt, so good job there. All right, the lipid bilayer. All right, so I introduced, I think it was yesterday, the phospholipid, which we have over there on the left-hand side. A phospholipid is a molecule that has a hydrophilic head, right? So these molecules here have a polar nature, so water is very friendly with them. And then they have hanging off of them 2 long fatty acid tails, right? And I said the other day that if we put these in solution and shake it around, what we end up with is we end up with a lipid monolayer, which looks like this. We have all the hydrophobic heads sort of pointing in the middle, and then we have our hydrophilic heads pointing out, right? So this is pointing out, this is in there, right? Well, a monolayer like that is a reasonably good way to move lipids around in the body, because essentially what we have is we have a little droplet then. Well, if we want to make a different environment inside than outside. You know, we talked about how that's fundamental for at least life on Earth. Okay, so if we're going to have that, well, we need to have more than a monolayer. We need to have 2 layers. And that's where the lipid bilayer comes in. The lipid bilayer, this structure, is the foundation for the cell membrane. It's also the foundation, in other words, how it's built, for the nuclear membrane as well. Now, the big difference between the nuclear membrane and the cellular membrane is the nuclear membrane is more porous. It allows more things in and out. The cell membrane is fairly resistant to molecules passing through it, at least molecules that are of a polar nature, which is most of the molecules that we find in the body. So this lipid bilayer, what we see here, this, with some additions, becomes the cell membrane. All right, so we have water on the inside, water on the outside, and then a barrier between them. Oh, that's a semi-permeable membrane. Okay, so this is why osmosis keeps playing a role in our physiology, because we have these semi-permeable membranes all over the place in all of our cells, in all of our nuclear membranes. All right, so if we zoom in and look at the one little section of the lipid bilayer here, you can sort of see the molecular shape. Now, what I'm pointing out here in the middle, these yellow structures, that's cholesterol. Most of the cholesterol in your body lives between the hydrophilic heads in cell membranes. Now, why is it there? Multiple reasons, but one of the characteristics it gives us is it makes the cell membrane much more fluid and flexible, right? So the cholesterol that's present helps to keep the cell membrane durable by becoming flexible. Now, I do want you to remember that even though this membrane is very fluid and loose, cells do have a skeleton, right? So their skeleton is going to determine their shape. But the cell membrane is the barrier between inside and outside. And our topic starting tomorrow is going to be membrane potential, which is all about the difference between the inside of the cell, the outside of the cell, and how the two interact. Okay, so that's why we talk about this. So cell membrane, phospholipids, cholesterol. All right, so sometime before tomorrow, give me 10 minutes outside of class and really look and learn this, all right? It will help you on Friday and Monday and, of course, on the exam that follows, right? So what we have here is on this side, so this is our fake cell, okay? This is our cell membrane. And we have a list of commonly found and commonly important molecules that are found either externally or internally. So we have extracellular fluid and intracellular fluid. All right, now we're going to further distinguish extracellular fluid as we go through the course, but for now we're at the cellular level. So we have ECF and ICF. What is present in those two compartments are very different from one another. Okay, so for example, sodium, lots of sodium in the extracellular fluid, very little sodium in the intracellular fluid. Big difference, right? Okay, so if I open a channel on the membrane that would allow sodium to pass, which way would it go? Into the cell or out of the cell? Into the cell. Why? Diffusion gradient, right? Concentration gradient. Diffusion is going to push it in. All right, so that's sodium, high outside, low inside. Potassium is reversed. Potassium in the extracellular fluid is very, very low. Potassium in the intracellular fluid is very, very high. So I'll ask you the same question. If I put a potassium channel on there and opened it, where would the potassium go, in or out? It would go out, right? Because it's going to follow its concentration gradient. Now we're going to make it a little more complicated starting tomorrow because both sodium and potassium carry a charge, right? positive charges repel, same charges repel, opposite charges attract, right? So we're gonna add the electricity card starting tomorrow. Okay, some other key ions. Calcium. That's a huge difference in order of magnitude, right? Look at all those zeros, 0.0001. That is a teeny, teeny, tiny, tiny, tiny little bit of calcium, right? How is that possible? Because the cell actively keeps calcium out of the intracellular fluid. And we're going to talk about a number of ways that it does that. But it does. And this huge concentration gradient, even though 2.4 isn't a very big number, compared to 0.0001, it's a gigantically huge number, right? So when we open a calcium channel on the membrane, well, by golly, calcium is going to go rushing in, right? It's down a waterfall of a concentration gradient. It's this big difference, one of the reasons that calcium is my favorite ion, right? Because it makes calcium twitchy. You open even a little calcium channel and you're going to get a bunch of calcium going in. That calcium is then going to make stuff happen. We're going to see that in nerve cells and muscle cells in particular. Okay, so those are the big three, the top ones: sodium, potassium, calcium. The others are less important, but we will talk about all of these as we go through the class. I will point out chloride. We almost never talk about anions, and you're going to find that in the next few weeks. The reason for that is chloride is the primary anion in the extracellular fluid, which you can see by the numbers there. So what's the primary anion in the intracellular fluid? Anybody know? Protein, believe it or not. Most proteins carry a negative charge. So the anionic component of the intracellular fluid is mostly protein-based. It's why chloride, we see it in the blood, we talk about it in the blood, but it has almost no relevance to physiology. We'd miss it if it weren't there, trust me. But you'd have to have a severe chloride deficiency to have any problems at all, right? So, and we'll talk more about that as we go. There's our friend bicarbonate. I told you bicarbonate's our primary base, or our primary buffer rather, our primary buffer in our physiology. You'll notice that there's much less inside the cell than outside. Let's skip down to glucose. Glucose inside of cells is typically quite low. And stop and think about why that might be for a minute. Why would glucose be low in the cell? Anybody want to answer? Because it's being used, right? It's inside the cell that we get glycolysis and oxidative phosphorylation. So the cell's own metabolism tends to keep the glucose inside of cells relatively low. Now, this concentration gradient works to our benefit because it means that if we have a hungry little cell, all it has to do is open a glucose channel and glucose will come right in. No active energy required to move that molecule, right? Now we get down to amino acids. Of course, amino acids much higher in the cell than in the outside, in the extracellular fluid, because it's inside the cell where all that fancy protein machinery is happening that we just saw the video for. Okay, we'll talk about lipids later. Down at the bottom, we have PO2 and PCO2. That's the way we talk about our gases, oxygen and CO2 as dissolved in fluid. And you'll see that our oxygen concentration is much lower in cells than it is outside. Similar to glucose, it's because we're using up oxygen all the time, right? Cells have a metabolism. That metabolism is fueled by oxidation. That's why we have an oxygen requirement. It's why we need oxygen in the air we breathe. CO2, it's the reverse. You know, every molecule of oxygen that the cell utilizes ends up becoming a CO2 molecule. So inside the cell, we have a lot more CO2 than we do outside of the cell. Now, interestingly, these concentration gradients for oxygen and CO2, you can even take it one step further and talk about the whole organism. So like every one of our cells needs oxygen. Well, how does that oxygen get to all the cells? There's much more oxygen in the air than there is in our cells. So we have a concentration gradient from air all the way into our cells. And that is literally how oxygen is moved around, is down its concentration gradient. CO2, same thing, right? We produce CO2 in our cells. There's much less CO2 in the air, so we have a concentration gradient that drives everything out of us, drives the CO2 out of us, right? Now, you'll notice that the concentration gradient for CO2 is not very big. That explains why we have a harder time getting rid of CO2 than we do in obtaining oxygen for our cells, right? And we'll talk a lot about that in the respiratory system, but we breathe 12 to 20 times a minute. The reason that we breathe at that rate is not to get oxygen. That's the rate we have to breathe to get rid of the CO2 that we produce, right? The only time we breathe for oxygen is if there's a lot less oxygen in the air, like if you're up high, for example. pH, remember that pH is a measure of hydrogen ion concentration, right? This one's reversed. The lower the pH, the higher the hydrogen ion concentration, right? And it's not a linear scale, it's an exponential scale. So what that means is the cell has a pH of about 7.0. The extracellular fluid has the same pH as blood, which is 7.4. So we once again have a concentration gradient, right? We have hydrogen ions are going to tend to leave the cell because there's more inside than there is outside. This also points out the fact that we are acid producers. All of the cells of the body have a tendency to produce excess hydrogen ions. So not only do we have carbon dioxide to get rid of, but we have our excess hydrogen ions to get rid of as well. And we will have a whole chapter together where we talk about the acid-base balance of the body. And all three of those at the bottom, PO2, PCO2, and pH, all of those have really profound clinical implications. You're going to take care of a lot of patients who have pH disturbances. You're going to take care of even more patients who have CO2 disturbances, right? So much more on that in the respiratory chapter. So, like I said, give me 10 minutes, make sure you know the difference between sodium, potassium, calcium, bicarbonate, glucose, PO2, PCO2, pH, okay? There's only 2 points on each of those graphs, so you can do it. I know you can. It will make your lives easier as we get into membrane potential next time. All right. So we do have the concept of membrane solubility. You could also call this concept, how easy is it for things to pass into a cell? All right? That the answer to that question is going to vary by what you're trying to pass into the cell. All right? So let's start with oxygen and carbon dioxide. Both of those molecules are nonpolar. All right? So what that means is that, here, let's go back to where's our bilayer? Yeah. All right, so both of those are nonpolar, which means they can pass through this membrane because the hydrophobic center doesn't affect them, right? So CO2 and oxygen, because they're very small molecules, they can slip between the hydrophilic heads, pass right through, and then right back on out. All right, so those are highly membrane soluble, is how we refer to that. And this helps us a great deal because it means that our cells don't have oxygen transporters or oxygen channels. They don't have CO2 channels, all right, because those substances can just pass right through the membrane. Now, that's the exception to the rule. Oh, the other exception are lipid-based hormones, like the steroid hormones. The steroid hormones are things like estrogen, progesterone, testosterone, androstenedione, which is an adrenal hormone, Cortisol is another. So you're going to hear about these in multiple classes. But the steroid hormones, because they are also nonpolar, they can do the same trick that oxygen and CO2 can, which is to pass right through the membrane without the need of a transporter. Now, I say there are exceptions because the vast majority of stuff that's moving around in the body is water-soluble. Water-soluble substances are polar substances. Okay, so if we go back to our lipid membrane, well, why can't a polar molecule pass through this? Well, it can pass through the first layer, the hydrophilic layer, but then it encounters this whole big region that is all nonpolar molecules, all those fatty acid tails pushing together. Well, a polar molecule cannot pass through that region. There isn't any way to stabilize it as it passes through, so it simply cannot pass. So this lipid bilayer is a wall to polar substances, to water-soluble substances. So if you've got a wall, the only way you're going to move through a wall, well, at least without big heavy tools, is you got to have a door of some kind. And that is how we have evolved. In our cell membrane, we have a variety of ways for water-soluble compounds to pass from the outside to the inside. Now, the thing with this system that's nice is it means the body, the cell, can very carefully control what goes in and what comes out. This is important for the cell's homeostasis. It's also important for the organism's homeostasis, right? So some of the ways we pass through, okay, we have channel proteins. Channel proteins are the simplest kind of door, all right? A channel is what I referred to when we were talking about sodium and potassium earlier. If I open a door that lets sodium through, well, where's it going to go? It's going to go down its concentration gradient. But it is going to pass through the membrane because we have a channel for it. Now, a nice thing about channels is they can be opened or closed, just like a door. So when the cell has a need for more glucose, for example, it opens the glucose door and glucose falls down its concentration gradient and falls into the cell, right? So that's a channel. Open and close, and the direction of motion is down the concentration gradient in a channel. So typically speaking, channels don't require energy to move the molecule. Some channels require energy to open or close, but they don't require energy to actually move stuff from outside in or inside out. Okay, so they're passive. Facilitated diffusion is another way that things pass through the membrane. And this is where a protein at the membrane, it's not quite like a door, it's more like an airlock or those revolving doors like they have at the airport. It's more like that. So it's not like it's open all the time. There isn't a path from outside in. But for the right molecule, if you step into the door and keep walking around the door, eventually you can get into the airport, right? So it works kind of the same way. By changing shape, it allows one or a small number of different similar molecules to pass through. I've got a better picture in a minute. Facilitated diffusion, because the molecule has to change shape, it has to move around the thing that's passing through it. These are saturable, like we talked about yesterday or the day before. If all of the revolving doors are running as fast as they can, more people are not going to get in the airport any faster, right? So we get a rate-limiting step where the most people you can get through that door depends on how fast the door is turning, right? So that's a facilitated diffusion. Another way we have of passing through the membrane is active transport. Now, as its name suggests, active transport does use energy, okay? And the reason it does is because this is moving a molecule up its concentration gradient. So, for example, sodium, if you look back at that page, sodium is high outside, low inside. Well, how does the cell get rid of excess sodium then? It can't just open a door and have it go down the concentration gradient, because if it opens the door, it's going to get a flood of sodium from the outside, which is going to make the problem even worse. So enter active transport. This is a pump, is one of the ways we refer to these. And what we do here is we use ATP, we make ATP into ADP, we use the energy that's released to push a molecule up its concentration gradient and inside or outside of the cell, depending on which way that we're going, right? So we also have limitations in active transport. It has all the limitations of facilitated diffusion, and it requires energy, right? So active transport is saturable as well. The only way you can speed up active transport is to make more active transporters, right? You've got to make more channels, which sometimes is what happens, as we'll talk about in the kidney chapter. All right, so if we kind of take these examples and put them in a conceptual picture, this is what's called the fluid mosaic model of the cell membrane. And the idea is you think of the lipid bilayer as kind of the surface of a lake or an ocean, right? Well, floating along in the surface of that are your membrane-associated proteins, the proteins that are going to accomplish things for the cell surrounding the membrane. All right, so we have some examples here. So here's our cell membrane: intracellular fluid, extracellular fluid, right? And then here's our lipid bilayer. Floating around like icebergs in the lipid bilayer are our membrane-associated proteins. So here's one example. This is a gated channel. So right now this channel is closed, but this, let's say a chemical signal arrives here and causes this channel to open. Well, then its inside is going to look more like this, and substances will be able to flow down their concentration gradient through this channel. See? And I think this is a great example of why protein shape plays such a big role in protein function. In order to have a channel work properly, well, it's got to be in the membrane, and it's got to have a hole through it that only allows the molecule of interest to pass through. And the vast majority of channel proteins are like that. So let's say this was a sodium channel, okay? Well, a sodium channel is not going to let potassium through. A sodium channel is not going to let calcium through. It's only going to let sodium through. See, so we have this selectivity, and it allows the cell to control the relative concentrations of things within it. All right, so that's a channel. We were just talking about that. Some others, so this would be an example of like an anchoring protein. So you'll see that this twisty protein right here is attached to the cell skeleton on the inside, and then it kind of reaches out. Well, what you don't see is it would be attached to a very similar protein on another cell, and this would hold two cells together, right? So that a tissue has some strength against pulling apart. We'll talk more about some specific kinds of cell-to-cell junctions when we get into the heart chapter a little bit later. We have recognition proteins. You're going to learn all about those in immunology, okay? Cells have to be able to identify each other and kind of communicate with touch, so to speak, between about what's happening. And it's the recognition proteins that make that happen. The MHC, you might remember the MHC proteins from previous physiology class. You'll learn more about those. Receptor proteins. A receptor is a protein that binds to a particular molecule, okay, and then changes itself in response to that binding so that the cell does something different, right? So for example, thyroid hormone has a receptor that's found on the nuclear membrane. Well, when thyroid hormone binds to that receptor, it triggers an increase in DNA translation and transcription, right? So it kind of fires the cell up a little bit. We're going to talk about a lot of different receptors this semester. So the ligand, just to clarify the word, the ligand is the molecule that attaches to the receptor that makes something happen. All right. Now, receptors, in order to do their job, have to be very specific. Like if you have a calcium receptor, it can't be activated by sodium, right? Or the system won't work right. So receptors have to be specific to a particular molecule. Carrier proteins are essentially any kind of protein that moves things across the membrane and the channels we talked about already. So this is just kind of a conceptual picture. Also, we tend to think of the cell membrane as being kind of like a smooth ball, because that's how we learned about it in our first cell biology class. It's not. The surface of a cell actually looks more like a forest. There's all kinds of molecules sticking up and forming somewhat complex shapes. And in my picture here, that's what we see with these glycolipids of the glycocalyx sticking up. It's mostly we think of it as smooth because we don't know a lot, so we just leave out the things we don't know. Every time we look closer at the cell membrane, we see more stuff. So it's a complicated region. As well it should be, because it's really defines physiology, that cell membrane does. Okay, so some examples. Here we've got channels, facilitated diffusion, and gates. Okay, so this is an example of a gated channel. In this case, it's a sodium channel. Here's our protein. When the gate is open, the protein changes shape so that this little flap sticks up instead of down. So when the channel is open, sodium can pass through, right? When it's closed, sodium can't. You'll also find in this channel, in this gated channel, that we have a lot of negativity on the inside. That serves two purposes. Number one, it helps the sodium to flow through faster, right, because it's being attracted by the negative, at least at the beginning. The other thing it does is it resists the passage of any negative ions then, right, because negatives repel negatives. So the sodium and potassium channels that we're going to talk quite a bit about starting tomorrow are sort of, they're opposite. They're installed in an opposite way, right? Because here the gate is on the outside, here the gate is on the inside. So the gate is usually on the high concentration side. And in terms of physics, that does make sense because you really wouldn't want a whole bunch of diffusion pressure sitting in the channel. So this keeps that pressure on the outside. Here's just a facilitated diffusion in a picture. Right, so we have our molecule of interest that comes in. It binds to this very specific binding site. So not any molecule will bind here, only a specific one will. When it binds, the chemistry of this binding actually causes the protein to change shape. So the entry closes and the exit opens. That change in shape then moves the binding site away. That releases the molecule, and now it's on the inside, right? So facilitated diffusion allows things to pass down their concentration gradient without energy, but we do still have saturability. That's a major player here. Like how fast it can go is going to be determined by how many of these proteins you have. All right. So we have a couple of different ways we can move molecules around. Right through here, A is diffusion, all right? So molecules moving down their concentration gradient. If they can, remember I said diffusion, you still have to have a pathway for things to move. The channel is the pathway that can either open or close, see? But as long as we have a concentration gradient, if there's a path, things will flow down that path. So that's A. B is electrostatically driven. And that's kind of a prelude to what we're going to start talking about tomorrow. But what we have in our picture here is we have a positively charged inside of the membrane, negatively charged outside of the membrane. So that means that molecules with a negative charge are going to tend to be attracted to the inside. Now, there's a limitation to that, as I'm sure some of you are starting to notice, and that is if we have all these negatives, will they just keep going in? No, because at some point there's enough negativity inside that it starts to repel more negatives. We're going to talk about that as the Nernst potential tomorrow, the concentration at which the electrical forces balance out the concentration gradient forces. Okay, so more of that then. Over here we have basic osmosis, right? If we have a lot of solute on one side, very little solute on the other, well, molecules are going to tend to move towards the area of high solute. All right, so that's osmosis. And then up here we have hydrostatics. This is our cardiovascular chapter, right? Because that's how the heart works. The heart creates a hydrostatic force, a squeezing force, which then moves whole cells around the body, right? Now, we have some other places where we see hydrostatics, but they all tend to be associated with the cardiovascular system. In the kidney, for example, our kidneys run on hydrostatic pressure. That's how they filter the blood. So we'll see that as quite a bit this year, or this semester, talking about the various organ systems. Okay, so the differences between active and co-transport. Well, active transport is the easiest one to understand, and that is where, here, let's find one. This would be an example of active transport. In active transport, we are using an ATP to push a molecule up its concentration gradient, right? That's active transport. Now, that's expensive for the cell. Spending an ATP to move one molecule, that's not the best plan, poor fuel economy, so to speak. So the body makes extensive use of co-transport to reduce its energy costs. All right, so what is co-transport? This is where we have two molecules that are moving through the same protein, either in the same direction or opposite directions. But here's the key to it. In co-transport, one molecule is moving down its concentration gradient. Another molecule is moving up its concentration gradient. Okay, so that's the key to co-transport. Okay, so for example, over here we have the sodium glucose transporter. This is a classic co-transport. It does not use ATP, all right? But we saw in our chart earlier that glucose is lower in the cell than it is outside the cell. Sodium is much higher outside the cell than inside the cell. So we have opposite concentration gradients, right? So in this case, what we have as sodium falls down its concentration gradient, it drives glucose, well, in this case, glucose is also going down. So here we have two molecules going down their concentration gradient. But that isn't always the case. Let's look at the sodium-calcium pump, for example. All right, sodium is falling down its concentration gradient. Do you remember the direction of the calcium gradient? In or out? In a big way, right? Here, let's go back and look. Come back here. Yeah. So calcium, very, very, very low inside, right? So that means calcium is going to have a tendency to flood into the cell if it can, right? All right, well, in this example, we're pushing calcium out. Well, how the heck? How are we doing that? How are we moving it against its concentration gradient without ATP? We're using sodium going down its concentration gradient to push calcium up its other one. My words got all messed up there, but sodium's falling down and pushing calcium up its concentration gradient, right? So this is useful because the body's not having to spend any energy. Now, technically, it has to spend the energy to make the concentration gradient in the first place. Yes?
So one doesn't always move down and one moves up.
They can both be going the same direction, but one is powering the other. So like in the case of the sodium glucose transporter, yes, glucose would come in on its own, but the sodium glucose transporter moves it faster, right? Good point out. Okay, hydrogen and sodium hydrogen transporter, here's another one. Hydrogen ion is moving with its concentration gradient. Sodium is also moving with its concentration gradient, right? So we have things moving in opposite directions. Probably the most important of the membrane proteins is the sodium-potassium pump. This is an active transporter. Technically, it's an active co-transporter. But what we get here, it's very efficient. This molecule moves 3 sodium ions out of the cell, 2 potassium ions into the cell, all for the cost of 1 ATP. Now, this singular protein is why potassium is high inside the cell and sodium is low inside the cell. This one protein is responsible for that. So when we talk about membrane potential next week, we'll see that the sodium-potassium pump, its job is to put everything back the way that we want it. In other words, high sodium outside, high potassium inside, so that we can make another action potential happen, which we'll talk about later. Okay, I'm out of time. Questions about anything?